Hi, welcome to another episode of Narcademy. In this particular video, we're gonna learn about the scientific method. The preview is seen on your screen. We're gonna start with the objectives of this lesson, and then we're gonna talk about simply what is really the scientific method and what are the different types of data and how do we collect them. And then we're going to learn about how hypotheses are formulated and tested. And then we're going to look at the theories, the difference between theories and models. And then lastly, we're going to summarize the entire steps of the scientific method. So what are the objectives of this particular video? We're going to know what's the purpose really of the scientific method? Why do we need to follow that in the field of science? And then we will be able to differentiate or distinguish what is a qualitative data and what is a quantitative observation. And then we're going to describe what is a hypothesis, what is a theory, and what is a model. Okay, so let us proceed. Scientific method is a logical approach to solving problems by doing observation. And then once you do observation, you're going to collect data. And from the data, you can formulate a hypothesis. And then once you formulate that hypothesis, you're going to test whether that hypothesis is correct or not. And then you're going to formulate theories that will support the data that you gathered. So, how do we observe and collect data? When we talk about observation, it is the use of our senses in order to gather information. So, when we observe, we're using our senses in order for us to gather data. So the data can be qualitative, which is a descriptive type of data, or quantitative, which is a numerical type of data. Now, what is qualitative? What is an example of a qualitative, and what is an example of a quantitative type of data? Now, that system is your basis for determining the data. Your system is your focus. It's the portion of matter in a given region of space that you select for focusing in the study when you do an investigation, an experiment, or observation. So that system, that is where you will gather your qualitative and or quantitative data. So let me give you an example of these particular types of data. On your screen, you see the difference between the two. When we say quantitative data, these are data that you got from your instruments, from your measuring instruments. These measurements or these instruments gather data that are numerical, meaning there are numbers, there are measurements. So some of these tools or instruments are rulers, balances, graduated cylinders, beakers, and thermometers, because these instruments have calibration, they have measurement values. On the other hand, if you look at qualitative data, these are the data that you use or that you gather when you use your senses to observe the results. For example, your sense of taste, your sense of smell, your sense of touch, your eyes, the sight, the sense of sight, and then the sense of hearing. When you use these senses, you're going to get descriptive or qualitative type of data. So I hope that you are able to distinguish between the two. So how do we now formulate hypotheses from our data? Scientists, they make generalizations or conclusions from the data given or data gathered. Scientists use the generalizations in order to formulate a hypothesis. A hypothesis is a statement that you can test. It's not a generic statement, but it's something that you can confirm or validate. So hypotheses are often in an if then statements format. So let me give you an example. How do we formulate an if-then statement? So if you look at the screen, there's a graph here. I want you to pause for a while and look at the meaning of the graph. Like what can be your possible if-then statements about your graph, about your data. Okay, so now you know that the x-axis is the time in days, and then your y-axis gives you or give us the growth or the height of the plant in centimeters. And there are four types of data here. The first, the yellow line, gives you the data for no fertilizer applied. The second, the blue line, is the 10% phosphorus fertilizer. And then you have the third, 25% phosphorus fertilizer. And then the last is 50% phosphorus fertilizer. So if you look at the data, when you are to make an if-then statement, you can say that for no fertilizer, you can say, if there's no fertilizer applied to the plant for 20 days, the maximum height of the plant is around 5 centimeter only. When you applied 10% of phosphorus fertilizer to the plant, then you will have a height of at most 10 centimeter of the plant. When you apply 25% of phosphorus fertilizer and at the same time 50% phosphorus fertilizer, the plant, those two plants applied with this amount, then will grow at most 25 centimeter growth. So the data clearly can give us a certain statement that will confirm a certain application, a certain validation of what we have done to the experiment. So by applying no fertilizer, 10%, 25%, and 50%. And you can also say that for 25% and 50%, there's no difference in the maximum height or maximum growth of the plants after 20 days. So that's how you formulate the statement. You analyze what data you have and then you make an if then statement and make sure that your statement can be tested or validated. Okay, so how do we now test the hypothesis? Testing a hypothesis will require you to experiment. You will do a laboratory. You will do a series of step-by-step procedure that will provide data to support or to devalidate or falsify a certain hypothesis or theory, making sure that the experiment has a very good design and an accurate method of gathering data. So we use controls. We use controls that will remain constant in the experiment. These controls are the ones that you can serve as a comparison to whatever variables that you're going to change in the experiment. So controls are the ones that you make constant. You don't do anything to it. Variables are the conditions that you will change in the experiment. So if you go back to our example a while ago, when you have controls, it means you have plants with no fertilizer. And then the variables are the setup or the plants wherein you apply the phosphorus fertilizer. Okay? So the phosphorus fertilizer can be 10%, 25%, or 50%. So the variable that you change there is the amount of fertilizer. The control is the one with no fertilizer. So how do we now formulate the theory after conducting the experiment? So when we have theorizing portion of the scientific method, we have two. We can have a model or a theory. So a model is more than a physical object. A theory is a statement that will explain the body of facts or phenomenon. So I will end with this particular example about a theory. So this is your model. There's an illustration that will explain the model of the atom. And this is the statement of an atomic theory that will generalize. So I will end there and please take notes. Bye bye for now.